Welcome back to our third video on Mendelian genetics, and so our fourth video on Mendelian genetics is what we're going to look at this time is Mendels' second law. Remember that last time we looked at Mendels' first law, the law of segregation, and this time we're going to look at Mendels' second law, the law of independent assortments. Let's get right into that. So with Mendels' second law, to reveal the second law from Mendels' experiments, we're going to look at a cross that involves two characteristics. That means there's going to be a gene for each characteristic. Remember, genes correspond to characteristics and alleles correspond to traits. So we're going to look at two characteristics, therefore two genes. And we're also going to then look at two traits for each characteristic. So the two characteristics are going to be P color and P morphology. So we're dealing with Mendels P plant still. So two characteristics, P color and P morphology. And then when we're dealing with two characteristics, we talk about doing a dihybrid cross because there are two genes involved and we're going to hybridize two plants. We're going to cross two plants together. So let's take a look at the characteristics and talk about the traits within the characteristics. So in terms of P color, P's can either be green or yellow. Okay, so we'll look at the gene which determines that in a moment. And then P's can either be smooth or wrinkly. So if you combine P color and P morphology, we can have P's which are yellow and round, like on the left. They can be yellow and wrinkly, such as kind of second from the left. They can be green and round, or smooth, I guess. And they can be green and wrinkly. So by looking at the behavior of the alleles for determining the two traits within these two characteristics, Mendel revealed the law of independent assortment. So like we did for the first law, the law of segregation, we're going to look at two crosses, and then in the next video we'll look at the explanation of these two crosses kind of at the level of meiosis. So let's take a look at the first cross to start with. So in Mendel's first cross, and let's just grab a pen very quickly here. So in Mendel's first cross, he's going to take a pea plant which has got peas that have the genotype of big Y, big Y, big R, big R. So let's talk about what the Y's and the R's mean in a moment. He's going to cross that with a plant which produces peas which have the genotype of little y, little y, little r, little r. So all of the cells in these plants are going to have the two genes, and each gene is going to have two alleles, obviously because it's diploid. And so we've got the Y gene, which is controlling P color, and then we can have big Y, which is going to specify yellow, and we can have small y, which is going to specify green. So big Y is dominant to little y. That means if we have a big Y in the genotype, we're going to see yellow peas. The only way we're going to see the green peas is if we have two recessive alleles, and that would be little y, little y. So that's the P color. In terms of the P morphology, the P's can either be smooth or round. Some people say round, we'll say smooth and round interchangeably, or they can be wrinkly. And that's determined by the R gene. And then the R gene also has two alleles, big R and little r. So for a P which is big R, it'll be round. And the only way we're going to ever see the wrinkly morphology is when we have two recessive alleles. So again, that means that big R is dominant to little r. That's always going to be the convention. So Mendel crossed a pea plant which was producing yellow, smooth peas. The genotype he knew from some other work he'd done was big Y, big Y, big R, big R. Now there are four alleles there. Remember, there are two genes, Y and R. And for each of those genes, there are two alleles. So we've got to deal with four alleles and potentially then four different traits, if you like. So we're going to do this cross here. The pea plant producing yellow, smooth peas, and the pea plant producing green, wrinkly peas. And so the genotype of that plant must have been little y, little r, little r. So as we think about this pea plant producing gametes via meiosis, each of the gametes can have one of the alleles from one of the genes. So the yellow, smooth pea-producing plant is going to make gametes, and each gamete is going to have a Y allele and an R allele. Can I only have one of each? Can I have two of each? Just one. And so because we've only got big Y's and big R's in the parent, the gamete of the parent can only have a big Y and a big R. And the same reasoning holds true for the other parent, and because that only has little y's and little r's, the gametes can only have little y and little r. Okay, so those are our two gametes. Now, if we do a fertilization event which brings these two gametes back together again, we end up with a plant that produces yellow round peas. And that's because we're bringing the big Y and the little y together to give us big Y, little y. And then we're bringing the big R and the little r together, and that's giving us big R, little r. And because the big Y is dominant to the little y, and the big R is dominant to the little r, we see the dominant phenotype, if you like, and that is yellowness and smoothness. And so we can explain the genotype and the phenotype of the F1 generation offspring from the P generation cross. So that's the first cross. Nothing really that we can conclude from that. It shows us some things about segregation. It was Mendels' second cross that becomes more kind of enlightening, if you like. So Mendel took his offspring from his first cross, and remember, all of the offspring are producing peas which are yellow and round. So what he does there is he takes those plants and he self-fertilizes them. So he literally takes a little paintbrush, and actually what he probably used was a little bee that had been stuck on the end of a stick. So he uses that, what's called a bee stick. He takes some pollen from the male reproductive parts of one flower and he brushes it on the female reproductive parts of another flower. So that's called self-pollination, and then when there's fertilization, that's called self-fertilization. So effectively is what he's doing there is a cross of big Y, big R, little r. That would be, for example, in the sperm of the plant, and then he's crossing that to the eggs in the plant, which will be big Y, big R, little r. They can't be anything else. That's what they've got to be. So if you think about meiosis occurring in these to produce the pollen and to produce the eggs, is what you can see is that you could have all these different combinations of alleles in the gametes, and rather than ending up with a 2x2 Punnett square, you end up by a 4x4. And so when we look at the genotypes of the parents, we can end up with gametes which are big Y, big R, big Y, little r, little y, big R, little y, little r. That'd be one parent and it's producing sperm, pollen containing sperm. And then the eggs would be the same genotypes as the sperm because it's the same plant, obviously. And so if you were to do this cross, and there's some controversy about whether Mendel's gardener was actually interfering with the experiment and using particular flowers when he did the crosses or not, but you never get these exact ratios that Mendel saw when you reproduce these experiments. But when you do these crosses, now when you do this self-fertilization cross, is what you end up with is an interesting phenotypic ratio. And so what you end up with is those 4, 5, 6, 7, 8, 9, you end up with 9 yellow round peas. Then you have 1, 2, 3 yellow wrinkly peas, 1, 2, 3 green smooth peas, and 1 green wrinkly pea. And this is the so-called 9:3 to 3:1 ratio. And the ratio is not that interesting and that important, except in that it shows us Mendels' second law. And so the second law basically states that the alleles of each of these genes segregate independently of one another. Now this only works if the genes are on different chromosomes. If you put both genes on the same chromosome, this doesn't work. So this only works for genes where they have loci on different chromosomes. So what does that mean? What does it mean when we say alleles segregate independently of one another? Is what it means is that it doesn't matter what one allele is doing. The other allele for the other gene will behave completely independently. That means you can take a big Y and put it with a big R during meiosis, or you can take a little y and put it with a big R. Or you could take a little y and a little r. It means you can make all the possible permutations that you possibly could. And if you think back to meiosis, that's because when you line up the homologous pairs of chromosomes during meiosis 1, you can line them up like this, or they could be the other way around. And that gives you all of the possible permutations. Now, Mendel didn't know about that, obviously, but what he did is he revealed that through this cross. And that's the insightful cross which leads to Mendels' second law. And that law is that alleles segregate independently of one another. So there's Mendels' second law. And next time we come back, we'll look at something a little more complicated and a little different. I'll see you soon.